Hello everyone, welcome to one more video of SPL BIO-TONIC, a channel to make biology subject easy. Students, in this session we will discuss about formation of pollen, its structure and development of male gametophyte. Students, in the beginning I request you all to like and share this video and hit the subscribe button to get notifications of all my new videos. Alright. Students, if we take a section of anther, it looks like this. As this anther is dithecous in nature, therefore it shows 4 sporangia. This transverse section shows 4 parts and all 4 parts are same. Therefore, one section of it is magnified and it looks like this one. Its outermost layer is epidermis. It is made up of flattened cells. The layer below epidermis is called endothecium. This endothecium is made up of radially elongated cells. Endothecium is followed by middle layer, which are composed of thin-walled cells. On its inner side, middle layer shows a single layer of cells called tapetum. This layer gives nourishment to the developing pollen. This tapetum also secretes sporopollenin. Students, we will talk about sporopollenin in the next video. This tapetum encloses pollen sac, which is also called sporangium. This sporangium contains microspore mother cells, which undergo meiosis to form microspores or pollens. These pollens develop partly in anther and partly on the stigma. In case of dithecous anthers, the 2 anther lobes are connected by connective, which we can see clearly in this diagram. Students, this diagram also shows lateral opening called stoma. This stoma is meant for the release of pollen during the dehiscence. Students, during the early development, anther develops from a single cell. This single cell is called Archysporial cell. This Archysporial cell divides by mitosis to form outer primary parietal cell and inner sporogenous cell. Sporogenous cell forms microspore mother cell, whereas outer parietal cell divides by mitosis to form wall of the anther. This wall of the anther, as we have discussed earlier, is composed of different layers such as epidermis, endothecium, middle layers and tapetum. Students, as discussed in the previous video, the tapetum encloses sporangium or pollen sac. It contains microspore mother cells which are diploid in nature. Here 2N means diploid. Each microspore mother cell undergo meiosis to form pollen tetrad, which in turn form 4 pollens. These pollens are haploid in nature. As at the end of this process, microspores are formed. Therefore, this entire process is called microsporogenesis. Students, these pollens may have rough surface or smooth surface. The pollens with rough surface are found in those plants which undergo pollination by biotic type of pollinating agents such as insects and birds. Smooth surface pollens are found in those plants which undergo pollination by abiotic agents such as wind or water. Now, let us talk about the transverse section of pollen. This pollen shows distinct nucleus and it is surrounded by cytoplasm. The wall of the pollen is called sporoderm. It is made up of 2 layers, outer exine and inner intine. Exine is made up of sporopollenin, which is secreted by tapetum. Sporopollenin is the most resistant chemical in the plant world. Therefore, these pollens are commonly found in fossils. Pollen shows some areas where exine is thin. These areas are called germ pore. The second cover is intine, which is made up of cellulose and pectin. So this pollen is broadly said to be unicellular, uninucleate, haploid, and covered by exine and entine. I normally call it as Panchasutri, that is 5 points. The branch of biology which studies pollen is called palynology. Please make a note of this. Now, let us continue with development of male gametophyte. This development takes place in 2 stages, partly in anther and partly on the stigma. While in the anther, pollen undergo first mitotic division to form large vegetative or tube cell and small generative cell. So here 2-celled pollen is formed. This 2-celled pollen is carried from anther to stigma. The second development occurs on the stigma. Once the pollen reaches to the stigma, the generative cell divides by mitosis to form 2 male gametes. It is called 3-cell stage now. That is the pollen with 3 cells. That is 1 tube cell and 2 male gametes. During the favorable condition, the entine comes out through germ pore to form pollen tube. The cytoplasm of the tube cell along with nucleus is also pushed into the pollen tube. Thus, by the end of pollen germination, male gametophyte is formed. This male gametophyte consists of empty pollen, pollen tube, 2 male gametes and tube nucleus. So students, I hope you are clear with formation of pollen, its structure and development of male gametophyte. In the next video, I'll discuss about gynoecium, structure of an ovule and development of female gametophyte. So stay home, stay safe. Thank you.